Chapter 6, Meselson and Stahl. To determine which model correctly matches and describe the process of DNA replication, organisms use Escherichia coli. Our DNA is made of these 4 bases in a variety of different order. They are the sole building block for our 6 billion bases genetic code that define our identity as a human or as an individual. But one important question lingered. When the cells divide, the daughter cell will have the same DNA sequence as the parental cell. How does this happen? In other words, how does DNA replicate? As you know, we all come from a single cell with having the same DNA sequence to an entire organism. And we are able to have children that share our DNA sequence because our eggs and sperm carries our DNA code. In 1953, the question lingered, how does DNA replicate? There were 3 theories. First was conservative DNA replication, followed by explosive and semi-conservative. The first one was conservative DNA replication, where the DNA molecule will get copied and produce an entire new second-rate DNA molecule. Second is the explosive DNA replication, where the DNA molecule will get cut at various parts and each will get copied and produce new rare DNA cut strand and will attach to produce 2 DNA molecules. The third one is semi-conservative DNA replication, where 2 DNA molecules will separate and each will serve as a template to copy a second new red strand. But which of these 3 theories was it? The only way to solve this mystery was to create an experiment. So, Matthew Meselson and Meselson Stahl decided to create an experiment to solve this mystery. Their finding was published in May 1958, entitled The Replication of DNA in Escherichia coli. They use bacteria because they can be easily grown in lab. First, they grow the cell in a medium of nitrogen of isotope N50, which is found in the DNA. This will make all the DNA very heavy. They use centrifugation, which could separate things according to their weight. Initially, all the DNA in the cell is heavy, where it was at the bottom of the tube, since it was grown in heavy nitrogen. Then, they started growing the cells in the presence of light nitrogen. After 1 cell division, the DNA was half heavy, so half of the DNA contained heavy nitrogen and half light nitrogen. This is not in line with the conservative DNA replication, which would predict that one DNA molecule is all light and the other is all heavy. However, this is in line with the semi-conservative DNA replication and the dispersive DNA replication model. After the second cell division, the DNA molecule is now either half heavy or half light or all light. This is now not in line with the dispersive DNA replication model, which will predict that the DNA molecule after 2 cell division will have the mixture of heavy and light DNA. Instead, this is in line with the semi-conservative DNA replication, which will predict that every cell will receive all the old DNA strands and 1 new one. Therefore, Meselson and staff discovered that the DNA would separate and provide the template for the production of two new DNA strands. Meselson and Stahl also identified the fundamental process where our DNA is passed on from cell to cell. This is the basis of inheritance and is an essential part for our development from a single cell to an entire organism. Thank you so much for your attention.